NIH to conduct global health research. And let me point out, of course, Brazil, which is our most significant South American partner, although we fund research in a few other South American countries. In fact, most of them, Brazil is the place where we have the largest collaborative connection, and we're very excited about that, and it's growing rapidly. I want to break my talk up then into telling you about 5 themes that are particularly high priorities for us at NIH, but they're broad themes, and when you look at them, you'll see that encompasses an awful lot of biomedical research. First of all, to apply breakthrough knowledge and technologies to understand biology and disease. This is a basic science component of what we're trying to do, which is these days often driven by new technological advances that make it possible to ask questions you might not have dared to ask even a few years ago. But then, of course, we don't want to leave that basic science sitting there. We want to translate that into better treatments, the translational science agenda. And going even further down, we would like to be sure that healthcare itself benefits from science, which means you need to continue the research enterprise, even in clinical situations where there's already the practice of medicine involved, but you may not be exactly sure what works and what doesn't. A fourth and very important agenda for me, since I've been the NIH director now for almost 5 years, is global health. We're part of a global community. We have resources and talents that don't necessarily live within country boundaries. We have needs for health that extend across the world. We should work together on these issues, and that's very much part of NIH's mission, represented here today by myself and Roger and all the others who have spent time in the U.S. who are sitting in this room. And then last, but really important, that most important resource, the biomedical research community, and in particular, we want to be sure that we're providing an opportunity for the next generation of scientists, many of them in this room, to have the opportunity to explore really interesting scientific questions and to have a path forward to explore their own creativity in independent careers. So I'm going to say a little bit about each one of these, not too much, and of course it's a challenge to give a talk in a place like this that only is going to last about an hour without sort of skipping over a lot of important things. So again, I'm going to choose some examples in each of these areas. Please don't think that that's intended to be exhaustive. And I've seen that in the posters presented this morning. The Human Genome Project, which I had the privilege of leading from 1993 to 2003, was an international effort, involved 6 countries, about 2,400 scientists, and ultimately succeeded in reading out that first reference sequence of a human genome. But now we'd really like to understand how to apply that in a way that benefits patients. And you can see from this cartoon that maybe it's not so easy. Just having a lot of A, C, G, and T in front of you when you're trying to solve a medical problem is several steps away from where you need to be. So together with our international partners, we went on from that first reference sequence to some other things, like trying to understand that roughly 0.1% of the genome that differs between individuals, the HapMap project. We went on now to sequence 1,000 genomes, actually it's more like 2,500 genomes, to high complexity and accuracy derived from individuals all over the world to try to get an understanding really about human variation. And we funded projects like ENCODE, which bring together, again, teams of scientists from multiple countries to understand how the genome functions, how it is that transcription is controlled, because, of course, only about 2% of the genome is coding for protein, and the rest of it is complicated but really important to understand if we're going to claim that we know what the genome is up to. And ENCODE has produced a lot of very interesting data. All of these projects designed to immediately release their data into the public domain with the idea that we want to empower the best and brightest minds wherever they are in the world to go faster by making these kinds of data sets immediately and freely accessible. Well, another consequence of that effort, which is causing quite a lot of interest and potential medical advances, although it turns out to be pretty complicated, is the use of that information about human variation to identify where the areas in the genome are that confer risk for common diseases. This very complicated looking picture, which is drawn upon a cartoon of the human chromosomes, 1 through 22, and the sex chromosomes. Each one of those colored circles represents a human genome variant. Most of these are single nucleotide polymorphisms that's relatively common in the population and confers a risk of a disease like diabetes or hypertension or cancer at very high statistical significance. These are not false positives, these are true positives. The breathtaking part of this is, if I'd shown you this diagram 6 years ago, there might have been a dozen colored circles over there. Now there are several thousand. And that's because the technology has made it possible. If you have thousands of cases of diabetes and thousands of controls who don't have diabetes, to sift through the genome quickly and find the roughly 82 places right now that are associated with risk of type 2 diabetes. Now those tend to be risk factors that are modest in their contribution. They only up the odds a little bit. But knowing that they're absolutely real, they're not statistical flukes, they point you to pathways involved in disease that you didn't know about before. In many ways, that is the power of genomics. It allows you to ask a question that's comprehensive. You're not limited by your prior hypotheses. You can ask questions that have the word all in them. What are all the variants that are associated with type 2 diabetes? What are all the genes that are expressed in the human beta cells in the pancreas? What are all of the ways in which chromatin, which reflects very much whether DNA is active or not, is organized in a particular cell type? The genome is a bounded set of information, which is fortunate for us. And we now have it within our sites. We're going to have a lot of work to do to sort out how it all works. But this potential of going beyond our guesses to really comprehensive analysis has been energizing and enlivening and has rocketed the field forward in many ways in terms of understanding the basics of how a cell functions. So all of those things are coming as a consequence of various technologies that followed on the heels of the Human Genome Project. The technology, though, that's probably going the fastest is sequencing itself. The cost of sequencing a human genome in September 2001 was roughly $100 million US. Look and see what's happened since then. This is a log curve, so this is actually showing you in remarkable ways how the cost has dropped now down to, according to this diagram, which comes from our US genome centers, something in the neighborhood of $5,000, but there is certainly a prediction that this year, based upon a new introduction of new instrumentation, the $1,000 genome, something that we thought we might ultimately achieve in another decade, is potentially here now. Now, of course, I should not overstate what that $1,000 genome means. That means that you've derived the nucleotides. It doesn't mean you understand it. The analysis is going to be a truly challenging effort, and is so today. One of the other things that technology has done, and if you've walked around the exhibit area, you've seen this, is machines that used to be the size of phone booths have now been greatly micro-scaled. So for instance, this is one of the DNA sequencing instruments. I'm holding it up in my hand here, about the size of a postage stamp, which is capable of doing whole genome sequencing. And who knows where this is going? Maybe nanopores will be the next big development. You can count on the fact that DNA sequencing is going to get faster and cheaper going forward for quite some time, and will advance probably more rapidly than Moore's law for computers, which, by the way, presents us with a bit of a big data problem, because all of this data being generated has to be stored, has to be analyzed, has to be made available in the cloud for all those individuals that have good ideas about how to use it. And there's something underway right now to try to deal with that called the Global Alliance for Genomic Health, that aims to try to at least establish what those standards ought to be for putting this data in the cloud in a way in which everyone can begin to learn from it. So how has this actually affected human medicine? Well, one of the areas where I think the greatest impact has been visible is cancer. Because cancer is a disease of the genome. It comes about because of mistakes in DNA, either inherited or more commonly acquired during life. And we have the potential now with this technology of being able to go much deeper in understanding what those driver mutations are in most of the common cancers that afflict people in the world. That's what the International Cancer Genome Consortium has been up to, seen here in a photograph of one of their more recent gatherings. So here the idea is to catalog all of the genomic abnormalities that you can find in tumors, hundreds of them from each tissue type, from 50 different of these cancer types and subtypes, and make the data available immediately to the research community so that people can begin to make sense out of it. And I have to say that it is a very international project indeed. You can't read the small print here of which countries are working on which tumors, but I will certainly outline for you one of them, which is Brazil. Brazil is part of the ICGC and is focusing specifically on melanoma, that most malignant form of skin cancer, trying to derive what in fact is driving it in patients with this disease. This, as you can see, is a research effort between several institutions, and the project is moving along nicely as part of this international collaboration. Another great example about how we can get together all of the bright brains that have dreams in a particular area and work on a team that will make this kind of information come along a lot sooner than it otherwise would. In terms of other uses of DNA sequencing, obviously trying to identify the causes of inherited illnesses has gotten more powerful when you can look at the entire genome and not just bits and pieces, and look and see what's happened here in terms of disorders for which we now have the molecular basis determined. Back in the late 1980s, when my lab was chasing the gene for cystic fibrosis, that kind of effort was something that took years and dozens of people, oftentimes with great frustration. Nowadays, with a DNA sequencer, some family DNA samples, a thermal cycler, you can answer those questions quite quickly, as you can see here. And in some instances, those kinds of DNA sequencing endeavors on rare diseases have had truly dramatic results. I'll tell you one little story here. These twins, Alexis and Noah, diagnosed with what was called cerebral palsy at age 2 because of their neurologic problems, which were apparent not right at birth, but after that. But they seem to get worse over time. Cerebral palsy is not supposed to get worse. Their mother questioned whether there was something else going on here, sought a new diagnosis. There was a clinical guess that maybe they had a form of a neurologic disorder called dystonia, and they were empirically begun on the same drug that's used to treat Parkinson's disease, namely L-DOPA, and they showed some improvement, both of them. But unfortunately, over the course of time, their response got less and less, and by age 14, these two twins had their health severely deteriorating, especially Alexis, who had had multiple visits to the emergency room because she couldn't breathe and was considered probably to be at the last step in what was clearly a very troubling downhill course. Well, being twins, obviously not identical twins, but twins with the same disorder, you'd have a strong suspicion, since both parents are completely normal neurologically, that this is a rare recessive disease. So a great opportunity then to use DNA sequencing to scan the whole genome and see if you can find the answer. Didn't fit any previously described disease. You only had this one family, but still, with DNA sequencing, maybe you can find the answer. So that was conducted. And basically, it identified an entirely new genetic disease, one which involved the production of neurotransmitters in the brain. And sure enough, one of the things these kids couldn't make was dopamine, but they also couldn't make serotonin. So by looking at where the block was in the pathway, they were started, in addition to the L-DOPA, on an amino acid, 5-hydroxytryptamine, that was distal to where their block was. And the results were really dramatic. Remember, these are kids who are pretty much at death's door. Within a week, you could see the response. Within a few months, that's what you see. Jumping on the trampoline. Alexis, who people were about ready to give up on, is now running varsity track at her high school. So once in a while, this kind of information points you to an intervention that is almost miraculous. I wish it was more often, though, because most of the time, you do the DNA analysis, you may find the answer, but it's many steps from there to coming up with a therapy that is going to help. But these kinds of stories certainly keep us all excited about the potential of where we're headed. Well, another basic science thing quickly to mention to you, and which was mentioned in the introduction, because I know there's a lot of interest in neuroscience in Brazil, and that is, it does seem like this is a unique moment for us to take all the tools that are being developed, the technologies, to try to understand the function of neurons, not just one at a time, but in circuits, and see whether we could mount this next moonshot for biomedicine. The BRAIN initiative, as it's being called in the U.S., but clearly this is also an international opportunity to try to learn the language of the brain. The President of the United States announced this just about a year ago. I had the privilege of introducing him And we have been basically now outlining what we think are the most important components of this. And in about a month, there will be more information about that forthcoming from the group that I've asked to assemble a more specific plan, including timetables, costs, deliverables, all of that. But we want to create a census of cell types in the brain. We don't actually know what those are. We want to figure out what the structural maps are. Develop network recording capabilities so you can see in that fourth dimension of time what's happening in the brain. Develop a suite of tools for manipulating the circuits so you're not just observing, you're also interfering. And obviously much of this will be done on experimental animals. We will want to be able to link neuronal activity to observable behavior. There will be a lot of needs for computation, theory, modeling, statistics. This is going to be a big data challenge. We will want to understand how imaging technologies that we currently use really work and how they can be improved. How we will collect human data, which you may be able to do, for instance, in the operating room for patients who are having epilepsy surgery, where it's generally possible to conduct temporary measures to see about brain function without doing harm. And, of course, make sure that we're developing an appropriate cohort of really knowledgeable and innovative neuroscientists. And this will mean an interdisciplinary effort, bringing together engineers, people who know something about electrophysiology, people who know a lot about how the brain works in the human, as well as computational biologists and so on. So it's a big agenda and a bold one. Sort of feels to me like the Genome Project felt in 1988, when it seemed like maybe it was a little too ambitious to talk about sequencing the genome. Well, maybe it seems a little ambitious right now to talk about understanding the human brain. Some of us think our brains aren't complicated enough to understand our brains. They might be right, I hope not. But it does seem like the moment has arrived, given the technologies that are emerging, and the importance of this for building a foundation upon which we hope we can place much of our hopes for understanding things like Alzheimer's, epilepsy, traumatic brain injury, schizophrenia, depression, autism, and so on. Okay, I spent a long time on the first theme. I'll spend less time on the others. Translating basic science into better treatments, though, is crucial if we really want to see those clinical advances. I showed you that already, and I mentioned a great example where the sequencing itself provided a treatment. But how are we doing in general? For those 5,000 plus diseases, how many of them actually have an approved treatment? It's pretty discouraging. The vast majority of those where we now know the molecular basis still lack an intervention. We have a lot of work to do here. And it's hard work. It's work that traditionally takes about 14 years and costs a very large sum of money, and the failure rate is enormously high. So many of us are looking at this as an engineer might, to say, is there a pipeline we can design here that's more efficient than what's been done in the past, where you go from a molecular understanding of a disease to an approved treatment more rapidly and more successfully? And one of the things that was done at NIH, which I'm actually quite excited about, is the formation of this new Center for Advancing Translational Sciences, which has that as its mission. It has multiple components, one of which is running our whole network of clinical and translational science centers around the country. Another of which is to take basic science discoveries that are kind of stuck and see whether it's possible to move them forward into clinical applications. So NCATS, as this is called, has a whole bunch of focuses on areas of rare and neglected diseases, including a collaboration with Brazilian investigators on Chagas disease, which is coming along pretty nicely. It's an opportunity to take what is commonly called the valley of death, where you have a great idea that might be clinically useful, but you get stuck along the way, and cross over that valley using the kind of talents and skills and resources that NCATS can present. Another thing NCATS is doing is to try to speed up this process of taking a promising molecule and actually getting it into a human clinical trial. And any of you who have done something about that will know that one of the things you have to demonstrate is that that molecule is not toxic. The way that's usually done is to give escalating doses to some small animals and some large animals. It's slow, it's expensive, and it isn't all that reliable, because you can find examples where the molecule seemed fine in a mouse or a dog, and yet when you gave it to a human, all of a sudden you had a bad liver reaction. So could we do better? Now that we have this amazing technology of induced pluripotent stem cells, that allow you to make human cells from any of us and then differentiate them into interesting tissues like liver or kidney or heart or neurons. Could you actually make a biochip, load it up with those differentiated cell types? Could you even make them not just living on flat plastic, but in a three-dimensional organoid, closer and closer to what you see in vivo? Maybe we could. In fact, we're working with 3D printing of cells to accomplish that. And then could you wire that chip up with all kinds of readouts to tell you whether the cells are happy or not? And then try your drug on it and see whether you get a signal that says all is well, or whether you see a signal that says, uh-oh, this one is going to be a problem. This is very ambitious. It's going to be another 2 or 3 years before we'll be able to say whether it works, but it's been really interesting watching the tissue engineering part of this develop. And ultimately, I'm guardedly optimistic, this may turn out to be a method for assessing drug toxicity that is both more accurate, cheaper, and much faster, amenable to high throughput than anything we have now. You can even imagine looking at individual differences in drug toxicity, what you'd call pharmacogenomics, because you can make a chip from hundreds of different people who have different genotypes. Think about that. I know, this is getting pretty far out there, but it's the kind of thing that we find pretty energizing in the current timetable. I have to tell you, as long as we're talking about taking basic science and bringing it forward translationally to benefit, that there is a particular application that the world is going to be electrified by on June 12th. And that is this ability to use the tools of engineering to design an exoskeleton for individuals who suffer from spinal cord injury and paraplegia, something which affects 6 million people in our country and many more worldwide. So in the opening ceremonies of the World Cup, and having only been here 24 hours, I can see the frenzy that's going on in Brazil about the World Cup, with probably billions of people watching. A young Brazilian paraplegic, yet to be chosen, I gather there were 8 or 9 who are in training for this, will, fitted out with this exoskeleton, kick the first ball. This has been work that's been going on for 20 years. It is led by Miguel Nicoleles at Duke, who's a Brazilian, and it is jointly supported by the U.S. and Brazil, including an NIH Director's Pioneer Award to Dr. Nicoleles. And this exoskeleton development has been supported by Brazilian funders, including FINEP. So this will be a moment, I think, for an audience that maybe doesn't think about science all that often, to see what exciting things are now possible. How does this work? Well, the paraplegic is fitted with a cap, which actually is able to detect brain waves. And so what the person is doing is to think about kicking the ball. And with appropriate training, it happens. It's even outfitted with sensors so that as the foot touches the ground, as this person is walking, there's a feedback so that that individual gets the sense, almost, as if they were feeling their legs again. Phenomenal work. Now, obviously, everybody's holding their breath, because this is really pushing it out there in a very public way. But the effort that's gone into this is remarkable and truly exciting. If you want to read more about it, there's a wonderful Scientific American story about this that you can find online. I have to point out that when we're talking about translational research, Science in its Breakthrough of the Year issue, which is always the last one of the year, this past year certainly highlighted a number of things that people in this room and people doing biomedical research around the world would be pretty excited about. In fact, our number one breakthrough of the year was cancer immunotherapy. Science usually picks things that are more basic. This is, in fact, an advance which is saving lives today. This is an effort to activate the immune system, which we've always knew was in there helping us out, fighting off cancer, but obviously when cancer gets really going, the immune system gets overwhelmed. What is it that you could do to reactivate that? So several new approaches. One is to block the protein receptors, to unblock the protein receptors that are blocked by cancer. So these are antibodies against things like CTLA4 and PD. The other thing, which is more technically demanding, but has given rise to some really dramatic cures, is to take the T cells out of the body for the individual, program them to attack a particular protein that you know is on the surface of those cancer cells and then put them back in. So-called chimeric antigen receptor, or CAR strategy. Again, this is a red-hot field for investigation, and one that is, I think, leading to lots of advances in a short period of time, even though we don't fully understand all the ways in which it's going to play out. Now, the rest of Science Magazine's breakthroughs of the year were, in fact, also ones we were pretty excited about, because, again, this is all of science, not just biomedical science. But in this case, 8 of the 10 were biomedical research projects, and things that we were invested in. Thank you very much. I just want to mention one more of those, and it's one that's also an effort that has been involving some Brazilian collaborations, and that is to design better vaccines. And particularly for HIV and for flu, and I'm going to talk about flu, we're learning by a combination of genomics and structural biology and immunology how to convince the immune system to be more clever than it naturally is, to fight off a virus that's very good at changing its coat. So influenza is covered over with this mushroom-sized shaped protein that you see here. This is the hemagglutinin. And the part of that protein that's most visible, and therefore leads to the immune system acting on it, is the sort of top of this mushroom cap here, the head. Unfortunately, it's the head that rapidly mutates year after year, which is why influenza vaccines have to be redone every year if they're going to expect them to have benefit. And of course, that doesn't help when you're thinking about that next worldwide pandemic, which we're overdue for, some H5N1 or H7N9 or whatever it is that's lurking out there for which we don't have current protection. What scientists have figured out is that if you look over the course of time, while the variations in that head protein are numerous and frequent, the stem of hemagglutinin is relatively constant. It sort of has to be, or the whole thing falls apart. And there are individuals, rare ones, who actually make antibodies against the stem, and those seem to be protective against all strains. So how could we convince the immune system of the average person to go after the stem instead of the head? Well, you can imagine strategies to do that by designing an antigen that is stem-specific, and perhaps convincing the immune system to go after that instead of its usual target. This has now reached the point of phase 1 clinical trials. I think there is a fair amount of optimism that this could be a reality in as little as 5 or 7 years. Obviously, though, with lots of clinical work still to do. In terms of healthcare itself, we don't want to stop at the point of coming up with a new strategy for clinical benefit. We also want to see what's already out there and is it working. And this is also an area that many research institutions are devoted to, and we are as well. And I'll just mention one example here, because it's one that involves a Brazilian collaborator. This is the study called the ischemia trial, which is an acronym. Everything has to be an acronym. I can't remember what it stands for, but it's ischemia. The I is, by the way, international. And it is an effort to try to understand, are we doing the right thing for people with heart disease, which is, after all, leading cause of death and disability worldwide. You can see the numbers there. So ischemia, oh, here it is. International Study of Comparative Health Effectiveness with Medical and Invasive Approaches. Isn't that clever? Led by one of our institutes, the National Heart, Lung, and Blood Institute, but it's a collaboration at 400 sites, 33 countries, and 24 of those sites are here in Brazil. The idea here, and here's the list of those investigators and sites, is to compare 2 initial treatment strategies for patients who have been diagnosed with coronary artery disease. A conservative approach where you treat with medical therapy and don't take on an invasive strategy right at the beginning, but only do so if medical therapy seems to be failing, versus an invasive approach where you carry out cardiac catheterization and revascularization right away. And across the world, some people are doing the first one and some are doing the second one, and we don't know what's the right answer for people who've just been diagnosed with this disease. We need to know. Here is a trial with very large numbers of patients in lots of different settings where we hope to sort that out. It's a great example of research in an area that is part of the standard of practice, but people have not agreed exactly what is the right standard of practice. You could call this comparative effectiveness research. Enrollment is scheduled between 2012 and 2016. Want to get 8,000 patients with at least moderate ischemia, 1,000 with chronic kidney disease. The fourth bullet, sharpening the focus on global health. Again, as I said, we're all part of one global community. We have health problems that are shared, some that are more geographically limited. We should be working on them together. And certainly, it's hard to point to a global health urgent issue more than HIV/AIDS. And we are making headway there, for sure. The development of highly effective antiretroviral therapy, saving many lives, research strategies that are controlling the epidemic by reducing transmission are being implemented, but we are still seeing way too many people infected each year. And we also need to do everything we can to prevent mother-to-child transmission, which is still happening in some parts of the world. And finally, we really need a vaccine to be able to stop this epidemic for all time. And that has been, of course, an enormously challenging effort. Although I would say, from my perspective, the promise of HIV vaccine research is looking better now than at any time in the last 30 years. In terms of treatment as prevention, which is a strategy which has certainly been one that has been controversial, I have to mention this particular trial, HBTN-052, which aimed to try to find out whether, in fact, by treating an individual who's HIV positive as early as possible, can you reduce the transmission to other individuals. So this study was basically focused on individuals who were in a stable relationship to see whether, in fact, treating the infected individual would reduce transmission to the HIV-negative partner. 13 sites around the world, including Brazil, 1,763 couples, and the results, dramatically published in the New England Journal of Medicine, showed that, in fact, early antiretroviral therapy, not waiting for CD4 counts to drop to a certain level, but right away, reduced the risk of transmission by 96%. Hailed by science 2 years earlier as the breakthrough of the year that year, and Brazil playing a very significant role in that effort. Another AIDS effort to try to reduce mother-to-child transmission, also worthy of note because of the importance of optimizing this. Ideally, of course, you want to treat the mother prior to delivery, but that still slips through the cracks. So what do you do when a baby is born to an HIV-positive mother and there's been no recognition or intervention? Well, there are ways to try to approach this. HBTN-040, again, an international effort, focusing on that. And in the course of study sites that you see listed here, making considerable progress, and I have to point out the sites mentioned, Brazil, Argentina, US, and South Africa. Brazil, by far the most successful in enrolling patients to participate in this study. These sites have done a great job in identifying, getting consent, and carrying out the study. And you can see already results coming from this to show, for instance, that 2 or 3 drugs are better than a single drug in treating infants who have just been born to mothers who are HIV positive. And of course, there are other infectious diseases that are a cause of global concern, and all of us should be worried about the fact that we have an increasing number of microbes for which our current antibiotics fail to provide benefit because they've developed resistance. I have to point out we had a problem with this in our own hospital in Bethesda, where we have the largest research hospital in the world. We had an outbreak of carbapenem-resistant Klebsiella, which was spreading amongst patients in the ICU, terribly ill patients. And it was only by a combination of complete genome sequencing of the organism and really good epidemiology that we figured out how that transmission was happening, and it was not what you would have expected. So we have to use the tools of genomics to be able to sort this out these days. But we also have to watch worldwide to see what's emerging. Again, it only takes one plane flight for one of these organisms to spread from one place to the other. And in this regard, I don't know if you saw, I certainly did just a month ago, this report in the New England Journal of Medicine about a particularly scary bug, a MRSA that has become vancomycin resistant, which was very carefully identified and characterized in a collaboration with investigators here in Brazil. listed, as you can see here. And this is one of those organisms that we all need to watch for, and we need better preparation for how to handle it. Well, finally, reinvigorating the biomedical research community. The thing that wakes me up at night is whether we are in fact doing all the right things to encourage the next generation of bright, talented, visionary, creative, ambitious scientists to be able to chase after those dreams. We certainly see this also as a global opportunity. I have to tell you that one of the things that I'm excited about being here to talk about is the relationship we have with Brazil in training. This NIH CNPq Visiting Fellows Program, supporting postdocs from Brazil who come to our intramural research program at NIH, which is about 17,000 scientists, and work in various laboratories, paid for half of their salary by Brazil, the other half by NIH, for 2 years, and then required to come back to basically spend time here to cover the cost of what was covered for them. We have already now something in the neighborhood of 40 to 80 of these postdocs on the NIH campus. They're terrifically talented. We'd love to have more of you. So, as well as, of course, many other universities in the U.S. that are participating in this program. Host you and be your mentor while you're there. I think it should be a great opportunity both for us to enjoy the talent of folks coming from Brazil to do great work in our laboratories, but also for people who come there from Brazil to be in the midst of a very exciting research environment, to be sure. If you want to know more about this, the website for Science Without Borders, which you see here, will tell you all about it. You can just browse around in there and you'll find various aspects of this program and how to apply. Well, I hope I've given you a bit of a sense from those 5 different areas of some of the things that we're excited about at NIH, and which I think this conference represents nicely in terms of areas of research that I've seen are being pursued here in Brazil and in other South American countries that are well represented here. And that's the way it should be. Science is not something that's sort of limited to one part of the world. Louis Pasteur said that very nicely. Science knows no country, because knowledge belongs to humanity and is the torch which illuminates the world. From being here, I can see that Brazil's torch is burning brighter, that the investments are being made here in terms of resources and training talent are paying off in exciting ways. And we at NIH look forward to continuing and strengthening our partnership with you at this most exciting time in biomedical research, where we really do have the opportunity to change the world. Thank you very much. Placed in the public domain. Obviously, that's not what happened with DNA sequences back in the 1990s. There was a veritable gold rush of people claiming bits and pieces of cDNAs as if somehow they had made some discovery that was worthy of a royalty payment. We were quite strongly opposed to that. I will say I was delighted by the Supreme Court decision in the United States earlier last year. In fact, there was one time I got to sit in the court and listen to the discussion amongst the judges and those who were debating the Myriad case. Of course, the Supreme Court basically decided, 9-0, which almost never happens, that a DNA sequence in its natural sequence of bases ought not to be patented. That simply the idea of isolating it without changing it in any way was insufficient to be able to claim any kind of composition of matter ownership. That has been a breath of fresh air into this otherwise tangled situation. Now, don't get me wrong. Patents serve an important purpose if what you're talking about is something that's much closer to a product and is going to require a lot of high-risk investment in order for a company to actually take that all the way to the point of something that the public can benefit from. And if you don't have patents in that situation, a lot of that development won't happen. But what we really think is you should limit that to things that are fairly far down the pipeline. And everything we're doing now with cancer genomics, for instance, is to put all that information immediately into the public domain. And once it's in the public domain, it counts as prior art, and it should therefore be patentable. We even have convinced, in the last 3 months, 10 pharmaceutical companies to work with NIH in a program called the Accelerating Medicines Partnership to try to identify the next generation of drug targets for Alzheimer's disease, diabetes, rheumatoid arthritis, and lupus. Normally, in the past, those kinds of target searches would have been considered confidential and would have been patented once the findings were obtained. But now it's all going to be in the public domain, open access, because it seems at this point we should just call this pre-competitive and we shouldn't get it all tangled up in these sort of obstacles. So I'm kind of where I think your question suggested Brazil is. Let's reserve patents for the circumstance where they make sense based upon public benefit. That ought to be the question. If this patent's going to benefit the public, do it. If it's not, don't do it.
Yeah, specifically, this decision was only in the genome or DNA products. But specifically, when you look at nature and isolate, for example, a plant extract, or a cell, or a lipid, or even a polypeptide.
Yeah.
And the decision, it really constrains the DNA, but the same aspects go to other nature products.
Yeah.
So United States, how is it going to deal in these terms? Is it going to apply this same decision to other products, or you think it's going to be restrictive to DNA?
So NIH has guidance for our researchers about basic discoveries of this sort, which would encourage them to be placed in the public domain. Because of a complicated piece of legislation that you probably know about, but most people in this room would not be familiar with, called the Bayh-Dole Act, all of the decisions about intellectual property for our grantees are taken by the institutions to which the grant was given. So if we give a grant to the University of Chicago and they make a discovery, they decide whether or not to file intellectual property for it. We have no say in that. But we can certainly say our opinion is the patent should be limited to those things that are more closely aligned to a product, but they don't necessarily have to follow that guidance. And that's going to take a little while to sort out. You have a good question there. I think you're right. The Supreme Court limited their decision to nucleic acids. The biotech industry, of course, is intensely interested in how this shakes out, and they would be quite alarmed if there was a sweeping decision about never being able to patent anything like a lipid or a protein that was derived from some approach. Again, our sense would be, unless you can make a case that you really need that patent in order to provide the incentive that's going to lead to a public benefit, it would be better to just make it available. But that's not an opinion that we can necessarily enforce.
Thank you.
I think we have time for a last question.
I'll change the subject. Back 60 or 55 years ago, when molecular biology was starting to become a new discipline, I have the feeling, because I am of that time, was easier to train a talented kid than now. Because we are dealing with a completely different biology. So, in your office in the NIH, is this a preoccupation?
So the last question, is it a...
In your office in the NIH, is this problem a preoccupation? How to train young kids, talented kids, to the modern biology?
That's a very appropriate question, and it certainly is something that we're constantly trying to optimize. We directly support in the U.S. about 1/3 of graduate students on grants that go to an institution for training specifically, or sometimes to that individual student. And there we have some control over what that training should include. And we try as best we can to be sure that that is responsive to all of the things that are coming along very quickly. So for instance, I don't think a graduate student in a research-intensive university should arrive at a doctoral degree without having some familiarity in computational biology. It is going to be so critical for so many of the things that we do in the future that you want somebody to be at least comfortable with approaching a large data set and not feeling like they're lost, but rather that they have some ideas about what to do. Similarly, I think we want to be sure that most of the trainees are comfortable with high-throughput nucleic acid analysis, because it finds its way into almost everything, not just genome sequencing, but even things that we used to think about would be done in other ways, like chromatin configurations, are done with a sequencer. We also, by the way, want to be sure that we're training our students to have a broad range of ideas about possible career paths. We've just come to the conclusion that in the U.S., many of our training programs assumed that all the graduate students and postdocs would end up in tenure-track academic positions. And the fact is, only about a quarter of them do. The others do other things. They go to industry, they end up in a primary teaching position, they work in universities, but not in a tenure-track position, more in a research associate professor position. And we have not really done a good job of letting people find out what is their own sweet spot. In fact, we've even set up circumstances where those who don't end up as tenured professors somehow assume that what they are ending up doing is second rate. We have to stop that, because all of these careers are critical for the function of this ecosystem. So yes, to answer your question, we are constantly trying to reevaluate the process. We have a harder time with the 70% of students who are actually being supported on a research grant through their principal investigator, because we can't quite reach in as easily and make sure that the training they're getting is appropriate for the way in which the field is evaluated. But we're thinking about ways to do that as well by asking institutions to be more clear about what kinds of training those folks are getting. The other thing that, just to finish, we have to pay attention to in the U.S. is that we are making students and postdocs take way too long before they get out into an independent position. And that is not good for the health of our enterprise. Many of the most creative ideas, going back through many decades that scientists have had, have not happened in their 50s, but maybe in their 30s or even their 20s. And yet most of the time, in the U.S. anyway, graduate students and postdocs remain in a position where they're responsive to somebody else's ideas for longer than I think is good for us. We need to keep the graduate program limited in number of time, and the postdocs should not go on beyond 3 or 4 years, and yet they often do. So we've got a bit of work to do there as well, and we're right all over it, recognizing that these are hard things to change overnight.
Excuse me, one question please, again.
Excuse me, if Dr. Khan
Okay,
accept a very quick question, I don't know if you could accept because he has a lot of things to do.
Thank you very much for the
Could you
nice
stand up please and give him
Okay. I think my questions are related to that from my colleague. The Brazilian Society of Biochemistry has a great number of young people, principally students. So I would like what you could suggest for these young people, the main suggestion to they target to with success and career. Thank you very much.
Well, as I understood the question, what is the best advice I could give for success? And boy, I could give a lot of ones, but I'll give just one. And that is choose an interesting problem. There's a lot of opportunities you will have, those of you who are not yet full professors, to decide what kind of a scientific problem you're going to face. You will be guided by others in your environment, but you'll have some choice about that. It may always be tempting to pick something where you're pretty sure you'll have a publishable result in 6 months' time. But sometimes that's not the right answer. Sometimes it's better to stand back, sort of take a little time, look at the field that you're going to be working in, and ask yourself, what are the really big questions that nobody has answered yet? And how could I get involved in answering those? That'll be a little risky. Some people will say, I don't know, you're kind of getting out there into an uncertain territory. But in the long run, that's how science makes advances. And that's how I think, if you're interested in contributing something that's really going to change our understanding of life, it's good to ask the questions that are most profoundly important. So think about that as a possible final exhortation I will have in the course of this hour. Thank you all very much for listening.